So I'd like to introduce our final undergrad finalist. This is the TU Munich team. Who's going to tell us about physical filter?
Fisco filter. Just 1 second. All right, so hello everyone. Welcome from Team TU Munich. I'm Katrin. Welcome to our presentation. So, clean water. It's the world's most important resource. But every day, tons of man-made pollutants enter the water cycle. Hormones, pesticides, and many industrial waste products all pile up in the environment and accumulate there, and they have potentially toxic effects. For example, hormones from birth control pills dangerously reduce the fertility of aquatic species. And these pollutants pile up because in common wastewater treatment plants, there's no sufficient way of degrading them. And because more advanced methods are expensive and energy intensive. We, the team from TU Munich, believe that a different approach to this global problem is needed. So in the past, many IGEM teams have used synthetic biology to remove pollutants from water. We want to use their ideas and build upon their ideas, but we want to improve their applicability by using a much more suitable chassis. The moss, Fiscomitrella patens. So our approach to fight global water pollution would be to use genetic moss that produces effectors for the degradation and binding of toxic pollutants. We believe that Fiscomitrella is an ideal chassis for such a project because of its self-sustaining, low maintenance and aquatic nature. It's a fully sequenced model organism and it offers a very easy and convenient and very efficient transformation procedure. So all these reasons make it a great chassis for synthetic biology. Just to show you how easy it is to work with Fisco, here's a sketch of the transformation procedure. You just have to linearize your DNA, enzymatically degrade the cell walls of the moss cell, and then mix it all together with PEG. Then you have to regenerate the cell walls for 9 days and select it on antibiotic plates for about 2 to 4 weeks. So we are actually aware of the fact that it takes much longer to work with a plant than, for example, with E. coli. So it's about 4 to 6 weeks from the transformation to the first experiment. But we believe that it's perfectly doable in the IGEM competition if you have good planning. And because Fiscomitrella is a natural part of the environment, it also offers other great application. So in short, we deliberately took the risk of working with this complex organism because we strongly believe that Fiscomitrella is the ideal chassis for such a biological filter and because we want to supply the IGEM community with this great organism. So let's get into the detail of the water pollution problem. Through many interviews with experts, we found out 2 main substance classes that are especially problematic. The first one being synthetic hormones from birth control pills, which, as I've mentioned before, disrupt the endocrine system of aquatic species. The second one being antibiotics, which increase selective pressure on bacteria and could thus increase the resistance of pathogenic bacteria. So with this class, we focus on the macrolides, which are highly persistent in the environment. So how do we want to solve these problems? We want to solve these problems by using the concept of bioremediation, which means that organisms break down hazardous substances into non-hazardous ones. And here we focus on 2 effector proteins. The first one being lacase from IGEM Team Bielefeld last year, which we used and which we converted to RFC25. But we also wanted to add a new effector to the registry, which is the erythromycin esterase B, in short, ERYB. It is able to break down the macrolide antibiotics, while the lacase is able to oxidize a very large amount of substances, for example, hormones. So to get to know the enzymes and to use the data we will generate in a model, we first made some enzymatic assays to get data on the kinetics and on pH and cell concentration optima. So for the lacase, we used the chromogenic substrate ABTS, and we found out that it has its pH optimum at pH 5 and that it doesn't like too much salt. So for area B, we did the same with the model ester P and PB, which also results in a colorful product. And here we found out that it likes higher pH values. But we also wanted to test this enzyme with the actual substrate, the antibiotic erythromycin. So we did what is called a Kirby-Bauer assay. We incubated our recombinant enzyme with the substrate and stopped the reaction at several time points. And then we applied it to filter paper and put this onto agar plates that had a sensitive bacterium on it. And here, the negative control, you can see that there's a zone of inhibition where the bacteria can grow. And as we expected, this zone of inhibition decreases with increasing incubation time with our recombinant protein. So we could actually degrade the actual substrate with our enzyme. But after all these in vitro experiments, we of course wanted to know, is our moss able to degrade it? So we incubated erythromycin for 1 hour with our transgenic moss, and then we analyzed the supernatant using liquid chromatography coupled to mass spectrometry. And as you can see here, for the wild-type moss, the initial peak remained totally unchanged, but for our transgenic moss, which expresses area B in the cytoplasm, you can see here that the initial peak has totally vanished and that 2 product peaks have appeared. So we can say, well, our Fisco filter works. We actually could degrade erythromycin with our moss. But since we do not only want to degrade antibiotics, but a very large variety of substances, we have to consider other parameters, like substrate, like if the pollutants can go through the membrane, or the availability of cofactors or disulfide bridges. So we need different localization systems. And so we made these 4 localization systems, which are cytosolic, secretory, membrane-bound, and the spycatcher-spytag system, which I'm going to talk about later. So I'm not going to further elaborate on cytoplasmic expression because it's straightforward. But to secrete our enzymes, we had to look for a signal peptide. And with the modeling software, we compared different signal peptides, and we found out that the Circ signal peptide from Fiscomedrella patents would work best, so we used this one. But we were not very happy about the secretion because we don't want to further pollute the environment with recombinant proteins. So we also searched for a membrane receptor to immobilize our effectors on the membrane. And we also did the modeling here and found out that we could also use the one from Fiscomedrella. And this is how our final construct would look like. And you can see here, on the one side, a GFP, which we will use to check for the localization and integration into the membrane. And on the other side, we here have a TEF cleavage site for easy removability of the effector. And for further experiments, we also included a strep tag. So to be able to prove that our different localizations worked, we introduced a new reporter protein, which is superior to other reporter proteins, the nano-look luciferase. And for example, we used the nano-luciferase to check for our secretion. And as you can see here, we checked for the luminescence in the supernatant, and we can see that for our signal peptide, the secretion is much higher than, for example, for the signal peptide Ig-kappa from mouse. So we could actually show in mouse that our secretion system works. And I'm also very happy to announce that we could show that our membrane localization works. This you can see very nicely in these epifluorescence microscopy images. On the right, you can see the wild type, and there's no fluorescence at all. In the middle, there's moss expressing GFP in the cytoplasm, and you can see that it's uniformly green. And on the right, we have our membrane-bound GFP, and you can see very clearly that the fluorescence is only around the edges. So we could prove that all our localizations worked, and then we could direct our effector proteins wherever we want them to be. So finally, I'm going to talk about a more advanced localization method. Because normally, if you want to fuse two proteins, you have the first protein and you can fuse the N terminals of the second protein only to the C terminals of the first one. So I'm very happy to announce for the first time in iGEM a system that enables non-canonical protein fusion, the SpyCatcher SpyTag system. It is based on a fibronectin binding protein from Streptococcus pyogenes that is split in a protein part and a peptide part. The protein part serves as a catcher while the peptide part is called the tag. So if you want, for example, C-C terminal fusion, you just have to add catcher and tag to respective C termini of your two proteins that you want to fuse. You add them together, the catcher recognizes the tag, and it forms an isopeptide bond between side residues. And so you have your C-C terminal protein fusion. But this is only one example how you could use the system. We think about like using this system for immobilizing multimeric proteins on the membrane, for example. So to prove the functionality of this construct, we fused the spy tag to YFP and the spy catcher to his tag. And then we incubated this for 1 hour, and we analyzed the result on a gel. And you can see here that it shows the functionality because we here have a fusion product that is the sum of the mass of the two parts that should be fused. But to further substantiate our results, we actually performed a pull-down experiment with beads that bind the his tag. And so this would remove YFP fluorescence from the supernatant because the his tag from the spy catcher is bound to the bead, and because the spy tag is fused to YFP, we should see a reduction of fluorescence. And we can actually see this in the supernatant. So we are very happy to supply the registry with this powerful tool. Now that I've told you what the Fisco filter is and how it works, my teammate Christopher is going to tell you how we are going to implement it.
Thank you. So, what have we learned so far? We've proved that we can localize our effectors wherever they are most efficient. Our effectors work in vitro, and the best part, our transgenic moss completely degraded the antibiotic erythromycin. So our Fisco filter really works. Now that we saw that the system works, we thought about its real-world implementation. To do this, we built a prototype of a closed tube reactor, which you can see on the left here, which allows an optimal area of contact between the contaminated water and the moss. However, upscaling this would be quite expensive. Therefore, we also considered two options for an open filter system. The first being an open channel system, where the water where the moss grows inside and the water simply flows through. The second option we considered, or we developed, is a triangular floating device, which we can see on the left here, where the moss grows on a felt cloth. And this can be placed on any water surface, both on wastewater treatment plants and on highly contaminated lakes or rivers. Because of its flexibility and low cost, this solution is ideal as for the large-scale implementation of our project. To monitor the system, we built an Arduino microcontroller, monitoring light, temperature, and water level, indicating, for example, whether the raft starts to sink. The data is then sent to a server via Wi-Fi, so the status of the raft can be easily monitored remotely. To determine the feasibility of our project, we traveled through Germany and visited several experts and conferences, since there were no experts on this topic at the TU Munich. And most importantly, we gained Professor Reski, the expert on working with Fiscomitrella patents, as an advisor for our team, and visited his lab in Freiburg several times to perform the moss transformations there. The company Greenovation, who use Fiscomitrella to produce pharmaceutical proteins and bioreactors, and the rest of our advisory board gave us a lot of helpful information and positive feedback on our project. Then we analyzed the economic potential of our Fisco filter, according to the aspects in this business model canvas. The result was that the Fisco filter is economically feasible, not only for commercial wastewater treatment, but also for water preparation in third world countries. The financing could be covered according to the polluter pays principle. So for example, when a new drug is developed, which costs around 1 billion euros, the company would also have to pay for the development of remediation enzymes, which would cost around like only 100,000 euros, so a fraction of the total cost, which could then be integrated into our MOS. If the initial costs were covered like this, the implementation price of our remediation rafts would be only about 50 euros, with next to no maintenance cost because of the autotrophic nature of Fuscomitrella. Now, while creating our business model, we realized that any new technology must be accepted by society in order to be successful. Through the great media interest in our project, we had already noticed that we were working on a relevant and hot topic. But additionally, we arranged several public events where we conducted a survey and discussed our ideas with the public. The result of the survey was that our filter system generally was very well accepted. But as we expected in Germany, which is quite skeptical towards GMOs, there were certain safety concerns. So to deal with the safety concerns of our genetically modified moss, we designed a kill switch. As a trigger, we wanted to use the red part of sunlight, since this allows activation of the kill switch without any human interaction. So our filter would be covered by a blue foil, under which the moss can easily grow. But when it leaves the filter, the full spectrum of the sunlight activates the kill switch and kills the moss. So for the suicide mechanism, we considered two options. The first was to use siRNA to inhibit a vital function of the moss, for example, photosynthesis. The second strategy we considered was to use to activate a nuclease which fragments the DNA and kills the moss in this way. For the siRNA model, we were able to prove mathematically that the vitality of the cell is reduced, but the cell is not killed off reliably due to a negative feedback loop. In contrast, for the nuclease model, the cell is always killed, the moss is always killed off effectively. So with these results, we designed the following light-triggered kill switch. We anchored a nuclease to the membrane with a TEF cleavage site in between. Additionally, a phytochrome B domain is expressed intracellularly, which is fused to a split TEF protease. Exposure to red light leads to a conformational change of the phytochrome B domain, allowing it to bind the PIF domain of a third fusion protein. This PIF domain is fused to the second half of the TEF protease, which is thus brought into spatial neighborhood of the first one, leading to the reconstitution of the TEF protease activity. The reconstituted TEF protease then cleaves the TEF site linker, releasing the nuclease, which is translocated into the nucleus due to its nuclear localization signal. There, it fragments the DNA and kills the moss. Now, we transformed moss with this kill switch construct, but unfortunately, when we opened the blue foil in the dark room, all the cells were dead. So the main goal is achieved. The cells are dead. A little too early, perhaps. We're currently investigating the reason and have already expressed the proteins for in vitro testing, but work is still in progress. On top of this kill switch mechanism, we proposed to use a non-sporulating strain of Fuscomitrella patens, which couldn't propagate in the environment, so that would give us some extra safety. Now, we also thought about extending our project to non-degradable pollutants. To do this, we've already transformed moss with PP1, which you heard about from Dundee before, who we collaborate with, to bind the toxin microcystine by simply binding it to transmembrane-bound PP1. Experiments are still in progress, so I hope to tell you more about this soon. Now, before we end, there are two more aspects which are important to us. The first is the auto-annotator, because when we planned our project, we looked at a lot of different biobricks, okay, and found that it was quite time-consuming to find basic information for some of them. So To change this, or to improve working with the registry, we've put together, or we've written a program, the auto annotator, which provides basic characteristics, which are listed here, for protein encoding biobricks, and returns them in a nicely standardized table. As a highlight, let's just look at the protein structural prediction plot shown at the bottom here, which shows the average hydrophobicity in blue and average charge in red, which allows you to immediately see transmembrane regions, which we have here, or stop transfer signals, which are there. We propose using this table as a new standard for protein encoding biobricks, and this proposition received the RFC number 97. Finally, because we had such a great time during this competition, we want to inspire and enthusiate a new generation of synthetic biologists. To do this, we put together educational kits containing explanations and equipment for some basic experiments, making bacteria glow and smell of banana. We then sent these kits to several schools and hope that the students enjoy the kits and become interested in the field of synthetic biology. Now, just to sum up what we have achieved so far, we've transformed and selected 23 transgenic moss lines and sent in a total of 73 biobricks to the registry. We verified the different cellular localizations of recombinant protein in the moss and proved the functionality of our effectors lacase and RGB in vitro and characterized their kinetics in detail. The best part, of course, our moss completely degraded the antibiotic erythromycin, so our Fisco filter works in the moss. Now, for the real-world implementation of our filter, we developed engineering concepts, including prototypes, and proved the economic feasibility of the filter. Then, most importantly, we successfully introduced Fiscomitrella patents as a chassis to iGEM, providing the necessary genetic parts to the registry. Now, I want to thank everybody who made this project possible, in particular Professor Skerra and his group who hosted us at their lab, and Professor Reski for his invaluable advice on Fiscomitrella. And I want to thank the TU Munich in general and our sponsors, Quiagen, Promega, etc. Thank you very much for listening.
Thank you. Thanks for the great talk.